The grades were low. The average was 71. The average on the first exam is usually 74 to 76. 25% Fs. I think that's a record. I saw it coming. I saw it coming. How did I see it coming? Attendance was down below 50% for the last 3 lectures. 70% of my questions are from that book, almost verbatim. 30% are things I say that are not in that book, things I say about that book. If you never come to class, if you make an A, you'll have to get it after class. If you make an A, you'll get a C. You know, so you really have to come to class. And so I think it was because of the high level of absenteeism. You guys are probably the ones that have been coming to class, so I have a question for you. How many of you didn't make the grade you thought you would have made? Okay? How many of you that just raised your hand are science majors? Okay? So you need to see me and learn how to study science. How many of you are not science majors? Okay, you need to see me. How to learn how to study science. The answer is black or white, yes or no. There's only one answer to every science question. There's almost no concepts. How you study for a science course is fundamentally different than how you study for a non-science course, where the answer is in the concept, the overview, the application, where if you're just familiar with the information, fairly familiar with the information, you can probably write yourself into a B. With science, if you're just familiar with the information, you're going to struggle to make a passing grade. Okay? If I had a how to study session next week, which would start out with you learning how you learn, okay? And you learning how, given your learning style, how you should be studying and how your brain works. If I did that, how many of you would come? Okay? I will set up an evening at a room. If you're not making the grade you want, you really need to come see it. It's not a reflection on your intelligence, probably not even a reflection on your motivation and desire. It's 100% reflection on after your 12 to 15 years of education so far, how many of you have been taught how to study, how to learn? And you can't pick it up like you do with a computer. You know, you have to learn it, learn how to do it. Most students that come to college don't know how to simply go to college. Go to class, and you don't know how to take notes. You can't even take notes right. I see you writing whole words and whole sentences. I ain't gonna cut it because I don't slow down. You know? I look at some of your notebooks and I see no notes. Whoa, that's even worse. So it starts out with coming to class, next is how to take notes, and then what we'll talk about next week is based on how your brain works, when do you need to study, and based on your learning style, how you need to study. So somebody has already told me that, whoa, I didn't make a very good grade on this exam. Wasn't quite ready for it. And they weren't saying I didn't study. They said I wasn't quite ready for it. But I had a friend who took the class last semester, and they did the same thing and ended up with A in the course. None of you are behind. Some of you have made a low enough grade where it's going to be your drop grade. But you all are starting at the beginning. And if you've done well on this exam, Probably means you've maybe just examined your way out of having to take the final exam. But every one of you have 4 exams left to make your grade. You make 2 low grades, another low grade on the second exam, then you're going to be in a hole that you have to dig yourself out of. Right now, I'm not worried about any one of you. What's my goal? What would make me happy? If you guys just pass. If I don't have to flunk anybody. 25% right now are sitting with the F. I'll tell you right now, I would guess I'm going to only fail 5% of you. The average is 4 to 7% every semester. And the ones that fail are going to be the ones that don't come to class or just give up, by and large. Cool. We did have some hundreds. You know? That was good. There is one question that I had to give 2 grades correct. The question on what part of the seed becomes the root. The answer was radical, but I also gave credit for hypocotyl. So there was one grade that was multi-keyed. On every question, I get a report on whether or not it is discriminatory, good, or bad. And I'll adjust it to make sure it's okay. All the other questions rated out as okay. Cool? So, I didn't mean to do that. How do I back out of that? Cancel? Thank you. Oh, you want to be able to see the overheads. Picky, picky, picky. Maybe I need to go how to lecture. I thought I hit that button. There it is. So we're going to have a little bit less than 2 lectures on hormones. Okay, we have about 2.5 lectures on temperature, a little bit less than 2 lectures on light, and then we're going to have exam 2. The first exam was riddled with a lot of new terminology and a lot of words. This exam has less of that. Typically the grades go up on exam number 2. Plus if you get your note-taking straight, your study timing straight, your how to study straight, wow, maybe we'll have 50% A's. If we have 50% A's, man, I don't know what I'll do. I'll have to give out prizes. I don't know, it might be a marshmallow, but a green tip banana, something like that. Okay, so that's our schedule for the next exam. Each exam is only covered since the last exam. So it's only the final that is comprehensive. So let's kill that. So let's talk about hormones. Hormone is the 3rd science, botany type background topic that we're going to cover. Then we'll get into the effects of environment. So we've already covered photosynthesis and respiration. Then we're going to do hormones. Hormones are what control the development of living organisms, plants and animals. How many hormones do humans have? I know my wife has 72. Must. Because whoo, she goes through some phases. She thinks I have 72. You know? Animal systems have a bunch. Plant systems are much more simple. Only 5. And in the beginning days of science, in the beginning days of academia, when humans started really studying how plants and animals grew, way back, they knew there were hormones in animals, that animals made chemicals that triggered reactions, you know, growth, development, mood, whatever. But plants were simple living organisms. They didn't think, they didn't move, they didn't reason. All they do is grow leaves and fruit. You know, so they're much simpler. And along came a fella that said, oh man, I think plants have hormones too. And the science of today said, you're stupid, you're crazy. a rebel. You don't know what you're talking about. He said, no, I'm pretty sure. Because what I noticed was, remember that doorway is my sun, right? Sun's coming up in the southern horizon, right? I lecture towards the south, right? So let's act like that doorway is a window. And I was standing right at the door. And I were a plant in a pot next to the window. What would I do?
Turn to lean towards it.
I will bend towards the window. The guy noticed that. Said, holy cow, that plant's moving. Yesterday it was this far. Today it's this far. It's moving. It's moving real slow, but it's moving. Wow. It must know there's light in that direction. So he turned it around. And what did it do after showing you his butt? It did that. Bent towards the window. And you notice no matter how you move that plant, it would always bend towards the window. He said that plant is moving with a purpose. Now I don't think it can think. I don't think it has nerves because we don't cut them open to find that stuff. There must be something that's making it perceive that light and grow towards that window so it can survive in nature. So he started playing with it. He was an experimenter. He observed some natural phenomenon in nature and then tried to investigate it. Okay? He noticed it was just the tip that moved, not the base of the plant. The tip and the young growth moved, but way down here it didn't move. So he said, something's happening up here. And he noticed the first thing to move was the very young tip. So he said, whoa, that's kind of cool. He cut the tip off, put it in the window. What did the plant do? Stayed right here. Did not move a lick. He said, oh, what does that tell me? Something in the tip is what's sensing the light and or triggering the movement, but it's something to do with the tip. So then he took some razor blades. Now they didn't really have razor blades like we knew them in those days, but they had thin sheets of mica. And he took the plant and he cut it halfway through. He didn't cut the tip off, he just cut halfway through and put a sheet of mica in there. So he cut it off, but he left this backside not cut. He said, hot dang. I'm not sure if the word dang was invented in the 1800s, but he probably said something equivalent of hot dang. Being the experimenter, he took another plant, cut it halfway through on the backside, put the same sheet of mica back there. It didn't bend. What did that tell him?
It has to be connected to move.
Has to be connected where to move? If he cut it here, it moved. If he cut it back here, it didn't move. What did that tell him? Something's moving, right? From the tip down. And it has to move down the back side, the dark side, not the light side. Okay? So what's the only way physically a plant can bend? I mean, I'm bending right now. What am I doing to bend this way? I'm contracting stomach muscles. I'm straightening out. What am I doing to straighten out? I'm contracting here. Or could I bend that way by elongating these muscles? Keeping these the same size but elongating these? Okay? He got a microscope and looked, and yeah, the cells on the backside got longer. When the plant bent, the cells on the backside got longer. When he put the mica back here, the cells did not get longer. And then he said, hot diggity dang. Something's produced at the tip of the plant. It's moving down the plant away from the tip to another location and causing an effect. It's causing the cells to elongate. And he said, by the way, folks, that is our definition of a hormone. A chemical in an animal that is made in one location, some certain organ or gland, and moves somewhere else and triggers some kind of developmental or physiological event. Plants got hormones. You crazy man. Plants don't have hormones. And he continued to study movement of plants. In fact, you can go to the library and buy a book called The Movement of Plants, and you will find that experiment in it. But also experiments on cucumber tendrils, waving in the wind and wrapping around wires. You know? Also, plants that open their flowers in the morning. Oh, they're so beautiful in the morning. They add glory to my life. That's a morning glory. A book on movement of plants. Who was that? Anybody know? It was the 1800s. Scientist way ahead of his times. First one to propose plants have hormones. Plants move purposely in their environment to get into a better situation for growing. They don't have nerves, they don't think, but they know how to adapt to their environment and survive. Who was that? Way before his time. You probably know him for turtles and birds. Charles Darwin. He did the same thing when he went to the Galapagos Islands and noticed the differences, okay? So that was Charles Darwin. He was the first one to speculate that there were hormones. Now, hormones are present in the plant and animals in incredibly small quantities. So he couldn't prove it. He couldn't extract and identify hormones. It took to the 1930s before the chemist extracted and identified the hormone that causes cell elongations of cells on the dark side of a stem to make them bend towards light, and it's called auxin, A-U-X-I-N. First hormone identified in plants. And since then, we've identified 5. Chemists and physiologists and biochemists have identified 5. So what we're going to do is we're going to go through each of those 5 hormones, talk a little bit about their function, where they're made, but mostly about what is it that can trigger a plant to do. Okay? You can talk to plants with hormones. You can spray them with a hormone, it will tell them to do what you tell it to do. So these are the things we use to manipulate plants so that they grow faster or prettier or whatever. Okay? So I'll do a little bit of science and a little bit of application. So the top, a hormone, an endogenous or naturally occurring compound, something made in the plant, made in one part, moves to another part, and triggers some kind of developmental or physiological event. Present in incredibly small quantities. As soon as we identified that hormone auxin, the chemists in the lab tried to make synthetic versions. Okay? The synthetic version is almost always more effective. Why? Hormones are very powerful molecules. Small amounts can trigger whole changes in plant development. Plants need to know how to make the hormone in the right place, in the right quantity, and when it's through, it has to know how to break the hormone down so it stops. If you put a naturally occurring hormone in a plant in an overdose, the plant will immediately start trying to degrade it. It will perceive, hey man, too much oxygen, let's start breaking it down enzymatically. So it won't be as effective. Synthetic version, you put it in the plant, triggers the response, but the enzymes, remember everything is made and broken down by an enzyme, there's not an enzyme that can grab that synthetic version and digest it, break it down, so they typically last longer. In nature, the naturally occurring hormone is indole acetic acid. Very often I will abbreviate IAA, indole acetic acid. The exam is multiple choice, right? You don't need to know how to spell it, just how to recognize it, IAA. And the synthetic versions that were immediately discovered by the chemists that acted like auxins are indole butyric acid, naphthalene acetic acid, and 2,4-D, 2,4-dichlorophenoxyacetic acid. Thank goodness I don't have to spell it, I just have to recognize it in a list. Which of the following is a synthetic auxin? The correct answer would be one of those. I may write out the whole word, I may give you the abbreviation. Okay? This is what it looks like. Simple structure, but I will never ask you a question about the structure. So why do I put the structure in the notes? I'm showing off. I'm showing, hey, I know chemistry. I took organic chemistry. I can draw that diagram. And they all are some version of that double string hexane. Where is it made? Shoot tips. We'll see in a minute that's really important on a lot of its effects. And in embryos of seeds. Well, the embryo of seed is a baby plant, right? So it's made in the seed too in the early stages of development. So know the naturally occurring version, know the synthetic version, know where it's made. Its main cellular effect is that it stimulates cell elongation. Its main cellular effect. Is that it increases cell elongation. It's the hormone that makes plants bend towards the light. It's the hormone that Darwin postulated fully 50 to 60 years earlier. Cell elongation. I'm going to show you a lot of effects. Whole plant effect, but at the cellular level, its main effect is cell elongation. So let's go to the next page. And let's talk about the effects. And some of these effects will have an application where you can use it. Okay? First of all, it is the hormone responsible for tropisms. What is a tropism? A tropism is a plant response to some type environmental or external stimulus. Charles Darwin was studying phototropism. Do plants bend towards light or away from light? Towards. So are they positively or negatively phototropic? They are positively phototropic. Guess what? Plants know up from down. You probably never thought about it, but why do stems grow up, roots grow down? You ever see a tree blown over? And what does the top of the tree do? The new growing points. Turn around and grow straight up. They can know when they're laying down, and they will grow up, and roots grow down. That is also an auxin response. If you have a tree in nature and it's growing vertical, the auxin is made in the shoot tip. It flows down all sides of the stem, causing the stem to elongate, so it grows up. Okay? If you lay that plant on its side, more of the auxin will start flowing on the underside than the top side. Those cells will elongate and it'll start growing up. Okay? So relative to geotropism, response to gravity, are plants positively or negatively geotropic? Or shoots positively or negatively geotropic? Negative. They grow away from the source of gravity. Now you take a root, lay it on the side, it grows down. More auxin accumulates up here and makes it grow down. Okay? Excuse me, more auxin grows here and makes it grow down, but it's kind of toxic, so it's a different thing. Bottom line is plants know where the light is and they know up from down. How many of you have been in the woods and ever looked up in the dense woods? What do you see? Leaves. Leaves, and nothing but leaves. And you may see sun sparkling through little bitty holes here and there. But by and large, in a dense woods, you will see no leaves. No sun. You'll see layers of leaves from the tall trees to the short trees. Go cut a tree down in the woods, let it fall. Now you have a donut hole. Sun coming in. Come back 2 years later, will the hole still be there? No, all the plants around go, hey, yippee-ki-yay, baby, light, 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 light. And they will fill up that environment and hog that space. Okay? That's the photo and geotropic response, going towards light and going up to keep capturing more light. Tallest tree doesn't worry about light. Redwood trees don't worry about light. Short shrubs on the floor, big time worried about light. When a hole appears, they start growing towards that hole. That's how small trees get to be big trees. Slow process. Thigmotropism, response to touch. Give me an example where a plant responds to touch. What?
Those little feathery leaves.
Oh, you mean the mimosa leaves, touch-me-nots? Fold their leaves? That is a response to touch. That's a thigmotropism. Venus flytrap closes when you touch it. That's a thigmotropism. Do you have any thigmotropisms in your garden? What?
Tomatoes.
Tomatoes, how are they phototropic?
They grow on the little stand.
Well, you have to tie them. You have to tie them to the little stems. Is there a plant that will cling to the little stems? Cucumbers will, what do they cling with? Those are called tendrils, okay? Cucumbers on the vine have tendrils, little wiry things. I wonder if we have a YouTube of that. That's a cucumber vine, and that's a tendril. Oh, wait, look at it. And that's what it's doing. It's sensed that pole, and it's stopping. And can you see when it flashes in the little curl? You know? You come back tomorrow, and the tendril will be wrapped. How many revolutions? If the tendril vine is doing this all day long, probably all night long. Waving in the wind, waiting to touch something. And then when it finally touches something, boop, triggers auction to form right there. Cells elongate. As it keeps touching, triggers auction to form right there. It wraps. How many RPDs? How many times does the tendril make a revolution per day? How many of you grow cucumbers? Your cucumber's not tied to the wire. You come back the next day and it's wrapped. How many wraps? 2 or 3. 2 or 3 wraps per day. So it moves very slow. You have to have a slow motion camera to get it. So plants can perceive their environment from touch also. They have touch genes that do that. Or tendrils positively or negative thigmotropic? Positive. When they touch, it triggers a response towards them. Oh, tulips. I just found out a year ago, my wife likes yellow tulips. These are just other kinds of movements. Most of the thigmotropic responses, auxin is involved. Okay? There's really no application of that other than that's cool stuff. I mean, you take a slow motion picture of that and calculate the RPDs, you know, maybe test different kinds of things it touches, see if they touch and wrap some things faster than the others, you know. What if you get it going and pull the pole away? Does it freak it out and does it unwrap? You know, you could do those kind of things, lay the plant over, see how fast it takes to grow up. You do that with your children, you'll win the state science fair. I'm serious, you will. You'll win the state science fair. Apical dominance is a biggie. Apical dominance. We talked about, I was going to start to erase that. We talked about the parts of stems and the buds at the tip of a stem. Yes?
Is the thigmotropism positive?
It's positive. Thigmotropism is positive. It's moving towards the touch. We had a stem, and at the tip of every stem is what?
Meristem.
It's a meristem, an apical meristem. It is big dog. You ever notice that a pine tree grows straight up? The main central stem is the most dominant stem. It has one tip, one stem going straight up. That's how we have telephone poles. The lateral branches are shorter and close to the tip. They don't grow hardly much at all. The apical meristem is dominating all the lateral meristems. The apical meristem is producing a lot of auxin, and it won't let the lateral meristems grow. So here's a picture. The apical bud is making auxin, it flows down, and that auxin prevents these lateral buds from growing. That's called apical dominance. On a tree, on a pine tree, apical dominance is incredibly strong. If you don't jack and kill the apical meristem, it will be just one central trunk. Make one big old tall telephone pole. Or one 300-foot high redwood tree. Oak trees on campus, do they have strong apical dominance? No, they have many trunks. Now, if you looked at an oak tree, each branch will start growing where it makes a single stem, and that apex will dominate the nodes for maybe a foot or two. Then eventually, one will start growing. When the apical meristem gets far enough away, it's kind of like your parents leaving home for the weekend. Yeah, yippee-ki-yay, baby, let's start partying! And they'll bust out of their buds and start growing. Okay? Plants grow by apical dominance. Strong apical dominance, one or just a couple of stems. Very weak apical dominance, just for a short period of time, and then the plant branches. What would happen if I cut off the tip of that branch right there? The auxin will stop coming down. Those buds will yell out yippee-ki-yay and take off growing. Every time you prune a branch, 2 to 3 to 4 buds will start growing. And as soon as they get some length to them, they'll make their own apical meristem, which then dominates that side branch. And if you want to branch more, you got to do what? Prune it again. The more you prune it, the more it branches. If I keep pruning it every year, I have a shrub. If I keep pruning it every year in the right location, man, I end up with Mickey Mouse at Disney World. Okay? All of our processes behind pruning are geared at overcoming or not overcoming apical dominance to force plants to grow in the direction we want them to grow.
Pruning is like actual removal, like cutting it off.
If you cut it all off, then nothing happens. Your tree's gone.
So it's not actually pruning.
Yeah. You know, if you have a limb and you cut the entire limb off, you remove the apical meristem and the lateral buds. The tree will never grow there again. Trees that have limbs growing towards my house, I kill their butts. I cut off all the way back to the trunk. I don't want them branching. You would prune just the tips. You'd probably prune it about a foot away from your house. Guess what? In 2 years, you're pruning the branches away from your house. So apical dominance and pruning go hand in hand. It's all related to auxin. Okay? Auxin flowing down the stem. I call it big dog. You know? This is big dog. You know, if you can't run with big dog, don't even get up on the porch, right? So my wife has 3 younger sisters. When we used to go home, they'd always spend their day shopping. Shop till they drop. And sometime I would go with them, but not shop with them. I'd watch them from a distance. And they looked like a bunch of ducks. My wife is in the lead. She's the oldest. And her little sister's right behind her. Store she goes in, the store they go in. You know? She got total dominance. If you take her out of the picture, they won't know which way to go. So apical dominant, big dog controlling everything. If you're a big dog pine tree, hey baby, I got you all the way to the ground. If you're an oak tree, oh, I got you for about 3 or 4 feet. If you're a yopon bush that branches like crazy, it's like, hey, I ain't got nothing up here, man. We can get about that much and that's it. Okay? Apical dominant. It's an auxin-mediated response. Basis of pruning. Okay, fruit set. Why do plants have fruit? Why do plants need fruit? Why do plants make fruit? To spread their seeds. And many fruit taste good, so people will eat, birds and animals will eat them and spread their seeds in their poop. Sometimes the fruit will be protective, like a coconut. coconut is fruit. It's to protect it, let it float around the Pacific Ocean for a couple of years, total protection. But the reason why plants have fruit is to make the seed and care for the seeds in some way. If you have a flower that isn't fertilized and it never starts making an embryo inside the seed, then the plant doesn't need the fruit. The plant only needs to make the fruit if there's a living seed growing inside. And plants can sense that. They sense that because the embryo of the growing seed makes a little bit of auxin, which diffuses out through the fruit, through the flower, and says, hey plant, I'm in here developing, don't get rid of this fruit. But if you take a plant and don't let it get pollinated, so the egg is never fertilized, so the embryo never starts developing, the plant will let the fruit start developing, and then when it doesn't sense auxin on the inside, it will abort it. And it will cause the fruit to fall off. Big problem growing plants in a greenhouse, especially in the wintertime. Tomatoes are pollinated by bees. If you have a tomato greenhouse and you're growing tomatoes in the wintertime, there's no bees to pollinate it. So you will make no fruit. So what do? You pollinate it. How do you pollinate it? You get a stick. You walk up to the tomato plant and you just, you don't hit it hard. You just kind of give it a little shake. And you vibrate in the flowers, making the pollen disperse. And you're hoping some will fall on the stigma of the pistil, you know. Or you can buy a little vibrator tool that's like a shaker. Okay? So we have to pollinate. It's not very good. You might shake the wire it's tied to, but that's not very good. It'll be enough pollination to where the fruit develops, but not a lot of seed inside. You ever buy a tomato in the middle of winter and it tastes kind of bland, not very gelatinous? Well, if you look really close, there's not many seeds in there. It's mostly fruit. And the flavor comes from the mucilage around the seeds. So you get a tomato, yeah. But it tastes like nothing. What does the seed make to tell the plant keep the fruit? Auxin. So what you do is you go to the garden center. You go to the garden center. Any garden center, including Wally World. And you buy a bottle, a can, spray bottle of tomato. I wish I would pay more tuition so we could buy faster computers. Why is that taking so long? I don't have the patience for you, website. There you go. Tomato blossom set. And you spray it. The instructions will be give 1 to 2 squirts per flower. Uh-oh, I picked the wrong one. Wrong active ingredients. I do need that one. Whoops! I've never got that one before. Let's try this one. Jungle juice. No, I don't want jungle freaking juice. That's fraternity parties. I'm not going any further down on Blossom set. We might meet Blossom. So I'm going to go with tomato. I always put a plant name in my search. That way I don't accidentally search stoma. There we go. Let me see if I can make it big. Big, big. Oh, I don't want to give you my email address either. Okay, you can't read the ingredients down here, but the active ingredient is an auxin. Typically they use NAA, naphthalene acetic acid. So you spray the plant with a small amount of auxin, it will make the plant think it has been fertilized, it has seeds developing, it will go ahead and develop the fruit. Still going to be mealy, not taste good, but you still have a tomato to sell during the winter time. Okay? Fruit or flower thinning. It's about to be cherry blossom time in the United States of America. And Lord knows, DC could use some color. Time in DC, around the mall. Oh yeah. There you go. There you go. That's what we're talking about. Those are cherry trees. Look at that cherry tree. Holy moly. How big are the flowers? They're about that big. How many flowers do you think they have on that cherry tree? Gazillion. If each one were pollinated, and very often that happens, each flower will start making a fruit. Okay? If each flower is that big, and each fruit gets to be that big, in the fall the tree would be 100% solid in cherries. But guess what? If it did that, the cherries would be little runts. Because the plant doesn't make enough photosynthase to make every cherry bigger. So what the plant will do is it will let its blossoms set after pollination. And it will let them start developing. But as soon as it perceives too many fruit on the tree, drawing on its reserves, it will start killing cherries. And it will cause extra cherries to drop off. Now, it might not seem such a big problem in cherries because the biggest cherry is not very big. But what if you're growing peaches in Texas? You know, if I had a peach tree in Texas, they would look exactly like that in the spring. What size peach do you buy? Size of your fist. You will not buy a peach smaller than that. You'll call it a runt. How many of you have natural peaches back home that maybe grew in your yard? They're always little runny peaches because you leave too many on the tree. So what peach growers have to do is, once the fruits start developing, once they pollinated them, start developing, they have to come in and remove the extra peaches. And they will leave one peach every 6 inches. If you leave about one peach every 6 inches, each peach will get the size of your fist, A-grade market quality. Okay? So if a plant perceives too many fruit, it will kill fruit. If you are a grower of fruit and you want market size, you have to go there and kill them. How do you think you knock the peach? Anybody here from a peach family with a peach orchard? How would you knock off the extra peaches? But this time I have to give it a whack. Wham! And I'm going to whack the whole limb to shake the tree. Or I can maybe have a shaker that shakes limbs, and I will physically shake the tree to thin them out. Or go in and hand thin. That gets to be kind of expensive. Well, if you take auxin and spray a high concentration, it will make the plant OD, overdose, and it will cause the extra fruit to fall off. So we can actually use an auxin spray to cause fruit or flower thinning, to cause the fruit or flowers to drop off. A very small concentration of auxin stimulates fruit set. A higher concentration of auxin stimulates fruit or flower thinning. And we do both to manage our fruit trees. And it takes the place of physical thinning and physical pollination. Now, we don't use it a lot because you got to have just the right concentration. If you knock too many fruit off, you affect your yield. So it's kind of dangerous. You got to know what the heck you're doing. So most growers will just resort to hand thinning or hitting the limbs with sticks. Okay? A little bit of auxin set, a little bit more auxin, a fruit drop. Herbicides. 2,4-D. How many of you ever used 2,4-D? What'd you use it for? Kill broadleaf plants. 2,4-D will kill all dicots, broadleaf plants. Very high concentrations of the auxin 2,4-D will OD the whole plant and kill the whole plant. So a little bit of auxin, fruit set, a little bit more auxin, fruit drop, a little bit more auxin, plant death. We're using auxin to manipulate. And then adventitious root formation. What is an adventitious root? Anybody knows?
The group that decides to go on its own.
Yeah, where would an adventitious root come from? Okay, so here's a little seedling on the left, and that's its natural root system. And this plant on the right is a cutting, where we cut off a stem and roots are coming out of the side of the stem to make a new root system. An adventitious root is a root from other than its normal point of origin. An adventitious root is a root from it other than its normal point of origin. Where do roots normally come from? Where do roots normally come from? Other roots. When you're walking across campus, do you see roots hanging off of leaves? Do you see roots hanging off of stems? Do you think there's roots hanging off of roots underground? Yeah, roots normally grow from roots. But if I cut a stem at the cut surface, it will make freaking roots. Why? Why would roots form on a stem where I cut it, but if I don't cut it, they'll never form there? What is accumulating at the cut surface to trigger the plant to make roots?
I would say auxin.
Auxin. Auxin is coming down the stem, accumulated at the cut surface because it can't keep going down. When you get enough of an auxin concentration, it will make adventitious roots. That is how plants know when their stems are cut off from the mother plant. You know, a hurricane comes through and blows limbs all over the place. You know what the limbs say? Thank you. I've been tired of growing on that old plant all my life. Now I'm over here, and if it's lucky enough to fall and land in the soil, where the end of the stem is below the soil, and it makes adventitious root, it starts a whole new plant. That's how some plants propagate themselves in nature, from pieces of themselves that break off. How many of you have relatives that in the kitchen window there's a bottle of water, and in it are cuttings stuck, forming little roots? That's what it is. The hormone that stimulates adventitious roots is auxin. You go to any garden center in town and go rooting hormone. A rooting hormone. Okay, here's a rooting hormone. A rooting hormone. Let's see if we can make it big. Boop, 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 boop. Ooh. Ooh, yeah. Garden safe take root rooting hormone. What's that word right there? Endobutyric acid. The active ingredient in rooting hormones would be auxin. This is the most common application of auxin we have, to treat our cuttings to make them form roots better, faster, and more vigorously. It's used commercially a lot. You can buy it in any garden center. Okay. It works on any stem and any leaf cutting. You mean I can take a leaf and I can make a leaf form roots? Let's see if we can find let's get out of Home Depot. Let's see if we can get back to Google. And let's see if we can find a rooted leaf. There's some leaves. Oh, this is my favorite. This is a rooted ficus leaf. It's a Ficus lyrata. Now, I've never rooted a Ficus lyrata, but I have rooted a Ficus decora. It's the one with the big leaves. And that leaf will form adventitious roots. That's it. You can pot that leaf up. Is it happy? Would it be happy? It would be happy. That leaf says, I got my own photosynthesis to make my own carbohydrates and everything I need. I got my own root system to make water and nutrients. I don't need anything else in the world. And you will have a plant that is nothing more than a rooted leaf. I was fascinated by that when I was a student. I used to root ficus leaves. And I'd put them in pots. I'd have pots of leaves. Dude, you got just a pot of leaves. Yeah, they're cool. You know? Call me weird. Hey, you want to go up to my room, see my rooted ficus leaves? I never could get a date. I don't know. But I was so proud of my rooted ficus leaves. On many plants, the stem will root and you generate the plant. Okay. And in tissue culture, what is tissue culture? Tissue culture is growing plants in the lab in usually glass or plastic containers and growing them in mass quantities. We can take a little piece of plant tissue. We literally can take a piece of a leaf, a piece of a stem, put it in tissue culture, and it will make little bitty blobs of callus. If in that tissue culture we add some auxin, the little blob of callus will make roots. If in tissue culture I can get some little stems to start forming from that callus, oh, here's a good example. I can get the blob and or the stem to start making roots. And I'm doing all this in a test tube. I can, in a simple petri dish, literally be growing hundreds of little plants from little blobs of tissue. And when I want them to make roots, I put some auxin in the tissue culture. And once the little blobs make plantlets and the plantlets to make roots, I pot them and I sell them to you. And we propagate plants that way, by tissue culture. We have a tissue culture nursery in Magnolia, Texas. I've been in one of their rooms about the size of this, and they will have racks in their tissue culture room. Loaded with rack after rack after rack, tissue culture room. We could have gotten in trouble on that one. Jars filled with little bitty plants, stacked up in rooms in little bitty jars. And there's a tray of them in a room. The manager was one of our former students, and I was in their room one day, and I said, April, how many little plants do you have in here? How many little baby miniature plants? She said, at any given 750,000. And they take them out of there and they pot them up like little seedlings. And they sell plants and propagate them in mass quantities. I can go steal a little blob of plant, go up to a leaf, little blob of plant, go into tissue culture and make 1 million of them, 1 million clones in 1 year. So in the animal world, cloning is very infrequent, kind of sometimes taboo. In the plant world, we've been doing it forever. Okay, so that's our auxin story. Let's go back to our table and let's look at the next hormone, cytokinin. Have you ever seen that word cytokinin before, or a version of that word before? Does it sound like a word maybe you've heard before? What does it sound like? Close. Cytokinesis, cell division. Cytokinin is the main hormone that triggers cell division. Auxin, the main cellular effect is cell elongation. Cytokinin, the main cell effect is cell division. When we first started tissue culture, we put plants in tubes. We'd put nutrients and water and all kind of stuff in there and sugar to substitute for photosynthesis, and they wouldn't grow very well. And then they found out if you put some coconut milk in the glass, it would start growing. The cells would start dividing. Well, what is a coconut? It's a big old fruit. And inside the coconut is a little embryo. In the coconut milk are all the hormones that a plant needs, including cytokinin. Embryos is one of the production of cytokinin. And then later on they identified the active ingredient in coconut is cytokinin. So when they identified the chemical, they named it after cell division. They knew it triggered cell division. And the naturally occurring form is zeatin, discovered in corn. The kinetin is also a natural version, but it's not discovered in corn, discovered in animal systems. Okay? The synthetic versions that we use typically are benzyl adenine, called BA. So if you want to use cytokinin, you're probably going to buy some BA. Where would you buy it? You buy it from the same place your high school science teacher buys their chemicals. Where is it made? In root tips and in embryos, and you don't need to know the structure. Now, what are some effects of cytokinin? Number one, it decreases leaf aging. You know, leaves have a certain life. When they first identified cytokinin, they'd start spraying it on plants and dropping it on plants and painting it on plants. And they noticed that when they put it on the leaves stayed green longer and didn't die as fast. So one thing it does is it delays leaf aging and therefore leaf abscission. Ooh, when we found that out, we got really excited. Why would I get excited about discovering a hormone that delays leaf drop?
Hopefully delays aging.
And why would I be excited about delaying aging?
Stay young forever.
Why would I be wanting the plant to stay younger forever? How many of you go buy plants within 6 months it's dead? And it's dead because all the leaves fall off. You got a naked stem there with a few leaves around the tip. If I could find the hormone for you to spray on the plant when none of the leaves fall off, I found the fountain of youth for plants, and you'll pay me a lot of money for that if your plants last longer inside. And it works. It'll keep the leaves greener for a full week or two. Well, yippee-ki-yay. Maybe one day we'll discover the one that lasts for a year. Right now, it's just a neat thing to know. Cytokinin will overcome seed dormancy and increase seed germination. If you ever go in a plant and you plant seeds and they're very slow to come up, try soaking them in cytokinin. Very likely it will stimulate them to germinate better. Okay. And then it favors adventitious shoot formation. A shoot is a stem. Auxin adventitious roots, cytokinin adventitious shoot formation. So let's see if we can find a begonia leaf cutting. A begonia leaf cutting. You can take a begonia leaf. See this begonia leaf laying on the ground? You lay it on the ground and you cut the vein. And every place you cut the vein, a little bitty adventitious begonia shoot would appear. You know, you take a cutting, you stick it in water, adventitious roots form. You take a begonia leaf and you lay it on the soil. Every place you cut it, adventitious shoots form. And if you treat that leaf with cytokinin, you can literally get hundreds of begonia shoots. It's very common. How many of you grow or know somebody that grows African violets? That's how they propagate their African violets. African violets. Propagation. That's how we propagate African violets. You take the African violet, you stick, you take the African violet and where did I go? Come on, where did I go? What did I do wrong? How do I get out of this new loop? Huh?
Go to the right,
Go to the right.
and your black, go all the way.
There. Ha ha ha. There it is. I got in trouble when I tried to make this bigger. That's an African violet leaf stuck in the soil like that, and you see that's little shoots coming out. If I take the cuttings and treat them with cytokinin, it may make more shoots. If you take a ficus leaf that has roots, treat it with cytokinin, you know what happens? Nothing. Nothing. You better be happy with just your ficus leaf. Yeah. And in tissue culture, we use cytokinins, shoot formation. If I type with one finger, I'd be fast. And tissue culture. We take those little blobs of callus and we treat them with cytokinin. It will make them make shoots. Gobs and gobs of shoots. You take the little shoots out and you pot them up in the soil like the little bitty plantlets. So what I'm really going to do is I'm going to have my blob of tissue in tissue culture. I'm going to let it grow into a big blob. I'm going to throw in some cytokinin and I'm going to make it make 100 shoots. Then I'm going to throw in some auxin, make it shoot, make roots. And then I take it out and plant it. So we use those two hormones in propagation. Adventitious root, adventitious shoot formation. You can go to the garden center and look for cytokinin and you won't find it. If you want to use it, you have to buy it from a chemical supply house, like where your high school teacher buys their stuff. Okay, so that's our cytokinin story. Not too many effects of cytokinin compared to auxin. In fact, auxin is the one with the greatest number of effects. So let's go back to our table. And our next hormone is gibberellic acid. There are over 120 gibberellic acids identified today. It's a very complex molecule. They differ just in a few side groups and side chains. You don't need to know the structure. There are no synthetic versions. We can make them, chemists can make them, but they're so expensive you can't afford it. So we use the naturally occurring version. We actually grow fungi that make certain gibberellic acids and we extract it from the fungi. So GA is the one example where we use a naturally occurring version in practice. Gibberellic acid's main effect is cell elongation. Now auxin was cell elongation too, right? Gibberellic acid is really big time cell elongation. It's even more effective. It is the hormone that triggers plants to truly elongate. So let's go back to our effects. It's made in the shoot tips, root tips, and embryos. By God, it appears to be made all over the place. And yes, it is. There are some plants that are dwarfs. We call them rosettes. They grow in a condensed rosette style fashion. Name me a plant that grows as a rosette. What kind of succulent? Can you think of the name of one? Aloe vera, sedum. Many succulents are rosettes. There's your succulents. Look at all those rosettes. Name me a vegetable that grows as a rosette. Well, let's just do vegetables. 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 That looks like some kale, looks like a carrot, looks like some spinach leaves. There's some chard. There's a funny looking little thing. There's broccoli, looks like some Brussels sprouts. There's some more kale. There's some more funny chef stuff. Oh, there's an ornamental kale. We don't eat it, it makes a nice little plant. Tazio, I don't know what the heck that is. Lettuce, cabbage, radish, carrots, rutabagas, mustard greens, collard greens, onions, garlic. They're all rosettes going close to the ground. When they flower, coming out of the rosette, flowering, let's try cabbage. Flower, I know there's supposed to be more than 2 weeks. We got kale. When they flower, bullcorn. Give me flowering carrot. Oh, there we go. There's carrot flowers coming out of the top. The Philly carrot flowers come out of the top of the carrot. That's called bolting. When a rosette flowers, the stalk gets long. And it looks like the flower bolts out of the rosette, so it's called bolting. Bolting is flowering in a rosette plant. Most rosette plants are biennials. A biennial is a plant that grows 2 years. First year it grows as a rosette, then it goes to a winter, and then the spring it bolts and flowers. Gibberellic acid is involved in that flower stalk formation. Gibberellic acid is involved in the bolting of biennials. It makes stems long. Now, I can also make a plant rosette by treating it with what we call growth retardants to block the plant from making gibberellic acid. So GA naturally makes stems elongated and biennials to bolt. If I block GA, I can make them shorter. And you will pay me a lot of money for that. Poinsettia treated with growth I can spell, I can't type. There you go. That's the same poinsettia. The one on the left is treated with a high concentration of growth retardant. That's as tall as it'll get. The one on the right is natural. Which one will you buy? How many of you would buy number 1 on the left? Number 2? Number 3? Number 4? No, you won't. You'll buy number 3. Number 4 in the store will look leggy, and you'll buy the one that's slightly more compact. Many of our florist quality plants, they are treated with growth retardants to make them about 2/3 height. They're more compact and more attractive, and you will pay more money for it. Doesn't cost any more to grow it, but a spray of growth retardant will allow me to take a $5.99 poinsettia and sell it to you for $12.99 through a florist. So we use growth retardants to make plants any height we want. We will continue next time. Thank y'all for coming. If you came in late and want your exam, please come get it.